Welcome, everyone. Thank you so much for coming to tonight's webinar. My name is Hillary Peterson. I'm the president of the Maine Entomological Society. I'm just going to give a quick update presentation on sort of the background of MES and why anybody would be interested in bugs. All right, so thank you to Bob Nelson for making this intro presentation. So what is the Maine Entomological Society? We are somewhere around 170 individuals from throughout the state of Maine, and also some from away, who are interested in the insects and other terrestrial arthropods like spiders, mites, ticks, etc., that are found in the state of Maine. We were founded in 1997 at what is now the Viles Arboretum in Augusta, and 10 of the original 14 founders are still active. We function as a common clearinghouse for information about Maine's amazing insect fauna. Most of our members are highly interested amateurs, including numerous Maine master naturalists. We provide a general forum for interaction and exchange of ideas. We do have professionals as members, but most professionals and academics are because of their work focused on very specific insect pests. Quote unquote, pest insects are maybe up to 75 of all of the species statewide, whereas the estimated total fauna in the state is 20,000 species. We are here to celebrate the biodiversity of insects. We are not a pest forum. We publish a quarterly newsletter with news and views on Maine insects. And those newsletters in the year prior to this one and all the way to the beginning are archived on our website. We also conduct monthly field days from late spring through the early fall and much more. And the newsletter has a table of contents, and the website also has our events. We also have a website, which includes an archive of those past webinar presentations, and webinars are scheduled throughout the winter month on various insect topics and are always free of charge to all members. We also have a Facebook page with over 6,200 followers called Maine Insects. It's sort of the identification forum and a nice little community. We're always trying to pull people into MES from that community. We've sponsored 13 entomological bioblitzes at an Acadia National Park. Members have been key players in 4 major faunal inventories in the state. And members have spent thousands of hours volunteering at the Maine State Museum. In 2022, we provided help in a project that resulted in May of 2023 of the pink-edged sulfur being designated as Maine's official state butterfly. And please consider donating. We have a scholarship fund that has active applications coming in, which is awesome. You can donate on the website or scan this QR here, or just take a picture of your screen. And then we have a couple of events coming up that I just want to show real quick. Not the right window. Okay. So on Saturday, as in 2 days away, we have the winter workshop. We have the amazing Ishvan Miko up again for that. And we're going to be doing some more on microhymenoptera. So on our website here, which is just maineentosociety.org, I have all the details. It's going to be held in Augusta and start at 9 a.m. and go until 2 p.m. I have parking information. I have a packing list on the site here. Any laptop or entomology supplies that you have, packing a brown bag lunch, reusable mug. There'll be coffee. If you want to bring a snack or something to share, you're welcome to. And we also are going to have a silent auction. So if you've got a bunch of piles of cluttered stuff that's entomology related that you're looking to offload, the silent auction, all proceeds will benefit the scholarship fund. And it really should be a fun day. Just from this picture, if you haven't met Ishvan, you can tell he's a pretty hilarious, pretty intelligent guy, really nice to spend time with and learn from. And then our next webinar on Thursday, March 14th is going to be Live Free and Pollinate, an investigation of native bees across New Hampshire apple orchard by Shiloh Favreau. So I'm going to briefly stop sharing my screen so I can introduce Ellie and let people in. Okie dokie. So, Ellie, we are so excited to have you tonight. I have a personal connection to Ellie. I first met Ellie in 2015, 14, 13, 2012 when I started, no, 2011 when I started at the University of Maine. In fact, I was trying to decide which college to go to, and Ellie gave me a tour of her labs when I was just thinking about UMaine, and that pretty much convinced me to go. So I go way back with Ellie, and she really provided me some of my first experiences in entomology, which I will forever appreciate. Ellie is currently Professor Emerita at UMaine, having worked as a professor of entomology for over 30 years before retiring in the fall of 2019. So I'm just going to share the screen with your stuff again, and just a little bit of ground rules, everybody. If you can stay muted and have your camera off just so bandwidth goes as far as it can, that would be fantastic. All right, let's share the screen again. All right, is everybody seeing Ellie's talk?
That's not the, I'm not seeing the first slide.
Oh, right, let's get back to that. There we go.
Okay, great. All right. And all right, well, thank you everybody for joining me this evening. And Hillary, it's a pleasure to talk to the Maine Entomological Society. I have been out of practice, having been retired for a few years, but hopefully it will go okay. Hillary, should I just have you advance the slides?
Yep, just say next and I'll just move it.
Okay, next. Okay, I think probably if you live in the state of Maine, you're familiar with the browntail moth, but for those few who may not be as familiar, like all moths, it's in the order Lepidoptera, and it's in the family Euribidae, and in the subfamily of the tussock moths. And another tussock moth that you might be familiar with is the spongy moth, which we used to call the gypsy moth, Lymantria dispar. It's in a different tribe, but it is closely related to gypsy moth or spongy moth, and they're both hairy caterpillars. Okay, next. And browntail moth was first reported introduced into North America. It's not a native species. It originates from Europe, and it's also found in the countries that border the eastern edge of Europe, as well as the northern coast of Africa. But it was first reported to be established in 1897 in Somerville, Massachusetts, in a location right next to a train yard, which may have facilitated its rapid spread throughout New England. It did spread fairly quickly from the late 1900s. It spread over about 15 years to its maximum range, which extended through much of New England into New Brunswick, Nova Scotia, down to Long Island. However, and it reached about 150,000 square kilometers. However, after that point, it slowly started to retract, such if you can see that by 1922, it seemed to be moving more coastal in its distribution. Next. And it continued to retract over the next 30 years, such that by 1973, when Paul Schaefer did his PhD dissertation on the population ecology of browntail moth in North America, he worked at the University of Maine. It was present at three locations in several islands in the Casco Bay. And there were a few webs that were found on inland sites. And then there were 5 discrete populations on Cape Cod. But all totaled, it was about 4.8 square kilometers in comparison to what it had spread initially. Okay, next. So from 4.8 kilometers, in 1973, it stayed very, very low up until the 1989. In 1989, there started to be small localized outbreaks of browntail moth, both on more islands in the Casco Bay and on some locations along the coast that abutted the islands. And but generally, from about 1989 to 2015, these outbreaks caused defoliation in the order of less than 10 square kilometers to 43 square kilometers. And it wasn't until 2016 when populations really shot up and we had a peak of over almost 51,000 acres or 206 square kilometers that were defoliated in 2017. That was the highest amount of defoliation that was recorded. But if you look to the right, this is the color picture is a picture of the 2023 winter web survey done by the Maine Forest Service, and which you can see on their browntail moth dashboard on their website. And browntail moth has continued to spread geographically over the last several years and is now found up to Millinocket and all the way east, heading down east, that might be by Machias and just about to the New Hampshire border. So its populations have been fluctuating in any one locality, but it is continuing to spread. Okay, next. Browntail moth causes defoliation, and so the caterpillars will strip their host plants. However, the main problem with this insect, as many of you probably know, is the public health issues. Browntail moth has urticating hairs that cause a mild to severe dermatitis when the hairs come in contact with your skin, either by the caterpillar coming in direct contact with the caterpillar, but also the hairs become airborne when they molt or when their cast skins fall to the ground surface, and you might be raking leaves or mowing the grass, and that will bring them back up into the air, and people can contact them. And there's a not insignificant proportion of the population that can have respiratory issues from inhalation of the spores. Okay, next. Okay, just a quick review of the life cycle of the browntail moth. Next. The moths emerge in early July. The adults, they're nice, beautiful white moths with the females have brown at the base of their abdomen, hence the brown tail. Next. And after dispersing, and they are strongly attracted to lights, in mating, the females lay their eggs on the undersides of the leaves. Next. And their eggs look like a little patch of fur because all those brown hairs that they have on their abdomen are used to cover their egg mass. Next. But you can see if you pull it apart, all the moth eggs inside. Next. So the eggs hatch in early August, and the small larvae begin to feed right where on the egg mass site, and they feed gregariously and kind of march their way down the leaf, feeding on the leaf surface and essentially skeletonize the leaf. Next. And you'll see, and it results in bronzing of the foliage when you have a lot of caterpillar feeding in the trees, and which is visible both from the ground and with aerial shots. Next. And as these small caterpillars move into September, they start to spin silk, and they cover themselves with sheets of silk. They have kind of a pre-web that they retreat back to as they venture out to feeding sites. And eventually they form layers and layers of this silk tying together leaves that they are feeding on in order to next create these winter hibernacula that they will next, which they'll basically spend from September through into April in. And these hibernacula attached to the twigs, usually at the very end of the branches next, at the very tip of their host plant trees. And you can see them when the sun is shining. If the sun is to your back and you're looking up at the trees on a very sunny day, you'll see these brilliant webs up in the trees, and you can distinguish them from other things by the silk that they wrap around the twig to attach it to the branch. Next. Now in the spring, when the caterpillars come out of their webs, they frequently emerge about in mid-April, and it's not uncommon for their host plant buds not to have started to swell yet, and you'll see the caterpillars resting outside of the webs. I live in Bodenham. We have a lot of browntail moth around here, and I look out at the trees in the spring, and the webs look like they've turned in olive brown. And it's because of olive greenish brown, because of the caterpillars resting on top of them. And you know that the caterpillars have emerged at that point. As the buds begin to swell, the caterpillars will extend a silk thread from their overwintered nest and will start to feed on the buds and follow the trails back to their web. They'll sometimes also build temporary new webs, spin them when in the spring as they're feeding, and they're still very small. Next. The caterpillars go into their webs at about the third instar, and they once they come out and continue to feed, they'll go through 3 to 4 additional instars as they mature, and they will begin to spread. As they get larger, they spread out on the tree to feed. And then next. Oh yes, wanted to say that this is the stage in which they produce the urticating hairs. So this is the dangerous stage from the time they emerge from overwintering through the time the moths emerge. Okay. And when they reach maturity, they form these pupal nests next. And the pupal nest is just a mass of loosely spun silk that wraps, kind of ties a leaf around them. And there can be anywhere from a single pupa to upwards, I think, I'm not sure what the maximum that we found. I know that I was just reading, Schaefer found up to almost 97, I think, pupae in one nest. So there can be a high number of pupae in a single nest at high population densities. They usually move down the tree a bit and pupate in the foliated range. Next. And then the cycle starts again when the moths emerge in early July. Next. So silk is really integral to browntail moth survival through much of the year. It guides them to feeding sites, both during the fall and in the spring when they emerge. It also provides them with a overwintering structure, which that protects them from the elements, also predators and parasitoids. And they also create a pupation nest that also protects them from predators and parasitoids. And you can see that, so that between that, most of the year they are involved with silk. Next. So this is a good time for me to just acknowledge the support that I had in my lab when I was working on browntail moth for about 5 years. not only the Maine Ag Experiment Station and the Northeast IPM Center, but also a number of local towns and individual organizations that were were being affected by browntail moth in the mid-coast and southern Maine that were interested in supporting this work. I had a number of collaborators and also a tremendous number of great undergraduate students who bravely worked with in the lab with browntail moth. Next. But in particular, I want to acknowledge the people who contributed to the work that I'm talking about this evening. Charlene Donahue, who I think most of you know, has been a close collaborator with me through all of my browntail moth work when she was working for the Maine Forest Service. And she was an instigator in getting us going on working with silk to begin with. Tom Schmilk, who is also with the Maine Forest Service, has taken over for Charlene and is leading the browntail moth work for them. Carla Boyd, who was my master's student, but well beyond being a master's student, she ran my lab and she was involved in every aspect of the browntail moth work that I was doing. And with the silk work, I had the opportunity to work with a number of great chemists, Dr. Barbara Cole and her postdoc at the time, Haewon Wang, who is now at the University of Lowell, and Dr. Carl Bishop also. And then the undergraduates who worked specifically on the web work that I'm going to talk about was Uwalina Nizam, who's worked in both my lab and in Barbara's lab, Katie Treblecock, who worked in my lab, and Josh Pico. And I'll talk more about their work as we go on. Next. Okay, so a little bit about silk. So silk is a protein fiber that's spun by a number of arthropods. And when we talk about spinning, we're not talking about braiding or weaving the silk filaments. It's really the insect or spider or organism processes aqueous protein solution into insoluble filament, which is extruded from their bodies. Next. All right, I'm going to focus on insects. There are a number of different insects that produce silk. In a review done by Sutherland et al. In 2010, they surmised that there were 23 independent evolutionary events across 17 orders that gave rise to the production of silk. And they came to this based on the type of gland that produced it, as well as and the location of the gland, as well as the type of protein that was also in the silk, and as well as the phylogenetic relationships. So the silk gland itself evolved from a secretory gland or cells that are located in multiple places in the body, but it's really the what is referred to as the type 3 dermal glands that evolved into producing silk, as well as Malpighian tubules and the labial glands. And as you can see, they believe that the use of those glands arose independently across multiple groups, as well as there was a variety, 14 different protein structures that evolved. I'm going to focus on moths, which the Lepidoptera, and as you can see that the Lepidoptera all have produced silk with their labial glands, which are their salivary glands. and have a specific type of protein structure that differs from other insects. They also, caddisflies are thought to have evolved with the Lepidoptera in the same mechanism for producing silk. Next. So insects use silk for a variety of different purposes, such as ballooning, which is a very common means of dispersal for a number of different caterpillars. Next. And that's a gypsy moth, and gypsy moth is that's the main means of dispersal for gypsy moth larvae. Also lacewings. Lacewings lay their eggs on silk threads in order to protect them from both cannibalism and predation. Next. And we have glowworms, Diptera, that produce strands of silk that are covered with sticky substances that glow in order to attract prey and capture prey. Next. And we have Lepidoptera, of course, who produce cocoons that are mostly consist of, in many cases, silk. Next. And then there are a number of insects, such as the eastern tent caterpillar, that produce webs, silk, in order to create these communal nests that protect them against predators, parasitoids, and pathogens. Okay, next. Okay, so most of what we know about silk production is we know from the abundant studies that have been done on the domesticated silk moth, Bombyx mori. And Bombyx mori, like the other Lepidoptera, as I said, have their silk gland, which is shown in this diagram here, is a paired gland. It's made out of 3 sections. The posterior region is where the protein, the fibrin, which is the protein strands, are created, as well as the saracen, which is used to coat them. And these materials are then stored in the mid-region of the salivary gland, and then they are extruded pushed into the anterior part of the gland, which you see is very narrow, and there's a drop in pH in this region, and the physical alignment of the strands happens, the fibers happen in this area, and a dense packing of the sheets and then the change in pH facilitates the establishment of hydrogen bonds that create a very strong structure that, when it comes out of the spinneret, is a silk fiber. And the silk fiber that we see is these fibrin strands coming from each that are coated with the saracen, which is the glue that holds the strands together. And in the Lepidoptera, these are labial glands, so they come out. The spinnerets are located in the mouth or the underside of the mouth of the caterpillar, as you see in the top diagram on the right. On the bottom diagram on the right is a longitudinal cross section of a silkworm, and it just shows how the whole entire body is permeated by the silk gland. The posterior part of the gland is located at in the latter part of the caterpillar body. The midsection is in the middle of the caterpillar body, and then the anterior section extends all the way from the abdomen of the caterpillar up through the thorax to the head. So it's been shown that about 20% of the body weight of the giant silk moth caterpillars is made up of their silk gland by the time they are ready to build the cocoon. Next. Okay, so when wanted to start looking at browntail moth silk. We were started by analyzing the microstructure of the silk, and this is work that Ewelina and Haywan did with Barbara and I. And Ewelina got some silk moth cocoons and harvested silk from both the cocoons and the browntail moth winter webs, cleaned them both mechanically and in distilled water, and then prepared them for scanning electron microscopy. Next. And here are some of the pictures. In the left side, you see the Bombyx mori, which is the silk moth, and on the right, you see browntail moth. And excuse me one second. And what I'd like to point out is on the top left, which shows the strand of the Bombyx mori silk. You can see it's a double strand of silk, and it's covered with the sericin and with some mineral deposits on it. And in comparison to browntail moth, what we noted with the browntail moth is that its strands are smooth, and they do not appear to be covered with the sericin, which is a striking difference and not something that we expected. And then the Ewelina and Haywan processed the silk, soaked it for 2 hours in hot water to try to dissolve some of the fibers. And what they saw was that they did get more fibers released in the silk moth prep. But if you look to the right, to the browntail moth, they observed almost more of a super contraction of the fibers, and it formed more like a hydrogel, and when they dried, it developed into a thick, very strong, papery material. So a very different behavior and appearance between the Bombyx mori silk and the browntail moth silk. Next. Now, the next thing that they did was to degum the silk, and they did this using sodium carbonate, which is a standard method for degumming, degumming is removing the sericin, releasing the silk fibers. And this is done in the production of the textile. So they clean the silk by removing the sericin in releasing the fibers to make the thread. And this had an impact on browntail moth. On the silk moth, you can see a lot more fibers being released. However, with browntail moth, they could not take any pictures because basically the sodium dicarbonate dissolved the fibers, and there were only just thin fibrils that were left remaining in solution that couldn't be collected. So again, a big difference between the behavior of the silk moth silk and the browntail moth silk. Next. And then this series of pictures that they took show a cross section of a piece of the silk cocoon and web. And you can see on the left with the silk moth that the silk fibers are laid down kind of almost randomly in no discernible pattern. Compared to browntail moth, you see these sheets of silk that are separated by airspace. So they're very organized sheets of silk with air in between, which, as you can imagine, would be a very useful thing for insulation in the winter. Next. Okay, so from the pictures of the microstructure, we do see that browntail moth silk is very different and from the silk moth silk. And Josh Pico did his honors thesis working with Carl Bishop and Barbara Cole in the last few years, looking at the amino acid content of the silk to see how that compares with other published work on amino acids in silk moths, amino acid composition of silk in silk moth. And in their study, what they did was to compare pre-overwintering webs with post-overwintering webs to see if through the process of wintering, the proteins change in the silk. Josh cleaned the silk and broke the silk proteins down in an acid solution, and then further processed the solution to generate a nuclear magnetic resonance solution, and which he ran through the NMR. When he did, it generates a spectrum like you see here in this graph, in which you have peaks that can be identified as specific amino acids. And Josh integrated the area underneath these peaks and compared them to one another, and that gives the relative amount of that amino acid present in the sample. Next. And this shows you a comparison of what Josh found compared to the silk moth, B. mori, and another wild silk moth, Genometa species that has gone through a similar analysis. And what you see, I've highlighted in yellow the 5 most abundant amino acids for each of the samples. And you can see that in Bombyx mori and in the wild silk moth, and this is consistent with some other wild silk moths, that the glycine, alanine, and serine are the most abundant amino acids present. And they are thought to form, these 3 amino acids are thought to form the backbone of the long chain in the fibrin protein. And there's a repeat of glycine, alanine, glycine, serine. And this amino acid sequence is thought to what gives a incredible strength to the silk and rigidity. And however, if you look at the browntail moth, you notice that browntail moth does not have, although glycine and alanine and serine in the pre-winter webs are the most abundant amino acids in the protein, silk protein, leucine is the most abundant. So there are some differences. And from what I've been able to understand in my reading about some of these silk proteins is that these, as I mentioned, the glycine, alanine, and serine form this backbone unit in the silk moth silk, and that consistency in that pattern gives it the strength, rigidity to the silk. Leucine and some of the arginine and some of the other amino acids have been described as part of the amorphous proteins in the silk. or then they're bulky amino acids, and it's thought that their presence lends to more extendability and less rigidity in the silk. And so this is suggesting that browntail moth silk may be less rigid than silk moth silk, and that lack of rigidity and more extendability may lend itself more to the type of web that they have, rather than the stiff cocoon that you see in the silk moths. Okay, next. Okay, so browntail moth silks differ from Bombyx mori silk. They don't have any sericin. Their fibers are also thinner than the silk moth, and they have uniform layers that separated by insulating space. The amino acid composition differs. That impacts protein structure and the behavior of the proteins, and probably affecting strength, elasticity versus rigidity, and also hydrophobicity. All right. So browntail moth, one thing we know is that the survival in these structures is very high. Next. When we, back one. So when we looked at the percent emerging from webs across different areas in infected areas in the state, really they generally ranged above 80%. I think it was a little lower in our sample in Bath, but it's upwards of 70%. So that's a very high overwintering survival. And that's consistent with what Paul Schaefer found back in the 70s. So these structures are incredibly strong. When you cut them open, you can see multiple layers of silk inside, forming a variety of little pockets inside between the leaves and the silk, and the caterpillars are nestled into these various pockets. And in addition to being well packed with silk, and some of you may have tried this, to pull these nests apart is actually very difficult. You have to cut them apart. So it is a very tightly woven structure. And the other thing that they do, if you look at the picture on the right, you can see these are the webs attached to the twigs, and they really extend the silk down. In some cases, the middle web there goes down over about 10 inches, more than 10 inches down the twig, that it is attached to the branch. So even though they are in the outer portions of the tree, they're not likely to be blown off. Next. Okay, so the caterpillars spend 7+ months through the winter months in these silk webs. So we asked ourselves, can we target them at this stage? They're out there for so long with something that would the caterpillars would be susceptible to, or could we disrupt the webs in a way that would make them more susceptible to our winter weather here in Maine? Next. So we conducted a series of trials to see if we could affect the integrity of the web. These were started in the winter of 2018 through 2019. We tested a variety of products. Our first trial, the webs were treated in the field in December. We used a handheld spray bottle, labeled the webs and sprayed them directly. And then we came back over every 3 to 4 weeks over the next several months and sampled them. So we had a sample over time. And when we collected them, we would put them in a cup in the lab and monitor for the emergence of the caterpillars. Next. And then we did a series of additional trials to increase. Overall, we did 10 products that we tested. We thought 4 to 6 of them could possibly disrupt the webs. They were detergents or other material oils that we thought could possibly disrupt the webs. And 6 of them were insecticides that may have had direct effects on the caterpillars. We in the subsequent, after the first trial, set up the first trial, in subsequent trials, we treated the webs in the lab. We came up with the help of my granddaughter, we came up with this great color coding system where we wrapped some of her pipe cleaners around the web to identify treatments and blocks and harvest dates for the webs. And we either brought them immediately into the lab and set them up for emergence, or we put them in an environmental chamber at 4 degrees C for 3 to 4 weeks, or we attach them to either trees, or in this case, they're attached to fences that are surrounding newly planted fruit trees in an orchard. Next, that's Alice. And then, so in the first trial, we found that of the materials that we looked at, the Safer Soap, the Turbo Spreader Sticker, we had an Orange Guard. Next, you can see the lowest bar, if the bar to the left is the control, and that the first 3 materials, we had as many caterpillars emerge as in the control. But with Orange Guard, which is a limonene, which is basically a material that's extracted from orange peel extract, it did reduce emergence of caterpillars significantly. Next. However, when we looked at the direct or indirect effects of limonene, next. In this graph, you see the blue is the control and the orange is the Orange Guard. And these are ones that were held in the allowed to emerge immediately in the lab, or in the cooler, or were at ambient conditions outside. And the fact that the Orange Guard was equally effective in all cases tells us that the Orange Guard was immediately killing the caterpillars, and it wasn't affecting the survival by impacting the web itself. If it was impacting the web, we would have expected to see caterpillars emerge in the lab. Okay, next. And so in the next series of assays that we did, again, we tested a variety of materials again. Next. Including Entrust, which is a spinosad, which is used for foliar treatments against brown-tailed moth. In that case, only the Orange Guard seemed to be effective. And next, but in the next set of trials, we had another material, pyganic, which is a organically certified pyrethrum that, when sprayed on the webs, also killed the caterpillars. Next. And in our the last materials that we evaluated, we had a rosemary oil was also as effective as Orange Guard. So we ended up with 3 materials, pyganic, rosemary oil, and Orange Guard, that all directly killed the caterpillars, but none of the materials we evaluated actually impacted survival of the caterpillars by impacting, disrupting the integrity of their webs. Next. Oh, rosemary oil. Okay, I wanted to bring your attention to, though, that in the third assay, one of the materials that we evaluated is this material Microtrol, which is another organically certified material, but it's the spores of an entomopathogen of insects, Bavaria bassiana, and the spores are formulated in a way that they can be sprayed like a regular insecticide. There are the spores there, they're grown in culture and formulated. And Microtrol did not reduce the emergence of the caterpillars coming out of the web. However, we held those caterpillars next following emergence. And what we did see is that 70% of the caterpillars that emerged from the mycotrol treatment died after they emerged. So they were picking up the infection as they came out through the web and resting on the web. And the spores would come in contact with the caterpillar. They would get infected and eventually die. Okay, next. And Katie Trevelcock also, who worked in my lab, did a really nice study where she evaluated 3 different species of nematodes, and she treated the webs with these commercially available nematodes. And she found that the heterorhabdited Bacteriophora was the most effective, but Steinonema carpocapsae also, another species, also reduced emerging caterpillars, whereas the one on the far right, Steinomena feltii, did not. This is particularly exciting because nematodes are commercially available. They're also known to be economically viable in terms of as a control means and a primary means of controlling grubs in turf and on golf courses. So this has some potential. One of the issues with nematodes is temperature tolerance. I don't think that the nematodes that are currently on the market would be able to be as effective in our April and early May temperatures as they were in this laboratory experiment that was conducted in the summer. But a number of several commercial providers of nematodes are working on selecting for low temperature tolerance in nematodes. So it could be something in the future. Next. Okay, so I've told you about our browntail moth web work. Is it a fortress that they have? A winter fortress? Next. Yes, definitely. They create a winter fortress that protects them from the elements. Next. Is it an Achilles heel? Next. Well, maybe. We'll see. It looks like there are some materials that are able to kill the caterpillars through the web, which may provide an opportunity for an alternative to having to cut every web out of a tree. And there may be some potential with these biological agents. Again, they would need to be tested more broadly in the field. Next. And with that, if you have any more information that you'd like to know about browntail moth, I would refer you to Tom Schmelk. Again, Charlene has retired. And so Tom would be the person to get in contact with the Maine Forest Service. Maine Forest Service also maintains an excellent source of information on browntail moth on their website. If you just go to Google Maine Forest Service browntail moth, you will come to it, and I recommend that. Also at the University of Maine now, Dr. Angela Meck is assistant professor of forest entomology, and she is continuing with various aspects of research on browntail moth and looking at ways to manage it for folks in the future. Next. And with that, I thank everybody for listening, and again, thank the people who have collaborated and contributed to this work. I did forget to mention Judy Collins. So Judy Collins helped us a lot with our spraying of winter webs. So with that, I would answer any questions if anybody has any. Thanks.
Awesome, Ellie. Thank you. That was great.
Okay.
We have one question in the chat. Bob asks or says, my understanding is that vireos are quite happy to go into the silk nest of tent caterpillars to munch through them. Will they also potentially go after the browntail moth caterpillars in their nests?
You know, there's a large list in the historic literature of bird predators of browntail moth. And in the 5 years that I studied them, I never saw a bird go after a nest. And I did see one time some orioles start to investigate, but they didn't pursue it. And then one time I saw some warblers actually all over. This was when they were just emerging from the nest. I saw some warblers all over this particular host plant. I think it was a cherry. And then when we went down to investigate it, we saw that there were winter moth all over that, and they were actually selectively feeding on the winter moth and avoiding the browntail moth. So yeah, I've never seen it. And I've torn apart tent caterpillar nests, and I can tear them apart. It's very difficult to tear apart browntail moth nests.
Great. Kathy Murray with a C. We have two Kathy Murrays. Asks, says, I have seen blue jays try to eat caterpillars in the web, but they seem to have difficulty. Karen Johnson asks, how does the nematode affect the browntail moth?
So the nematodes are interesting critters. What they do is they enter through either the spherical, the mouth, or the anus. So they have to go through a natural opening. And once they penetrate inside the caterpillar, they release a bacteria. And the bacteria very rapidly kills the insect, whether or not it's a caterpillar or a grub. It very active within 48 hours, usually the insect dies. And then the nematode lives on that bacteria. The caterpillar's body becomes kind of like a petri dish to grow the bacteria for the nematode, and the nematode feeds on the bacteria. And then when the nematode leaves the host, it carries the bacteria in a special vesicle in its gut, so that when it invades the next host, it can infect them.
Cool. All right. And then Kathy Murray with a K asks, do you think that the orange guard penetrates the webs, or does it just persist on the outside of the web where the caterpillars encounter it when they emerge from the web?
No, I think it does. I think the volatiles in the orange guard penetrate the web. I don't think it's just when they emerge, because we really get no emergence. So unlike with the mycotrol where they emerged and then died, with the orange guard, they didn't emerge. So yeah, I don't think that it's, I think that and the rosemary oil and the pygienic must be able to penetrate the web enough, and the volatiles from those materials kill them.
I'd be curious. So I did part of my PhD work as I looked at how trees respond to the stink bug glue, the eggs. And I'd be curious if any host plants respond at all to the web on the branches or touching the buds or the leaves. Because I just remember you said it was D-limonene. That was one of the substances that actually changed in response to the stink bugs laying eggs. So I wonder if there's any plants that would defend themselves effectively at all against the webs being in the branches.
That's an interesting question. You know, the leaves are being rolled up late in the season, and I wonder if a plant is more responsive to that type of manipulation late, you know, when the leaves are actively growing, than it would be late in the season.
Yeah, that makes sense.
Those leaves are going to desist and drop off the tree. So That would just be my first guess.
Yeah.
But it's an interesting question.
Kathy Marie with a K asks, oh, actually, wait. Oh, we have Kathy. Okay. Kathy Marie with a C asks, how broad spectrum are the chemical and biological agents you tried? In other words, what other insects would be compromised by their use?
Right. So the both of them are broad spectrum. Both the Bavaria, it attacks a number of different insects, and the nematodes also can infect a number of different insects. One thing about the nematodes is that they don't survive very long outside of a some type of protection from the elements. So they can, unlike the Bavaria, where the spores will just sit on the outside of the web, and whatever substrate they land on, they'll also break down quickly. But it might be that when you spray a web, it gets into the cracks and crevices of the web enough that it protects like the silk is protecting the caterpillars, it might be protecting the spores of the fungus as well. The nematodes actually actively burrow into the web and to hunt for the caterpillars and infect them. So one of our thoughts with treating these nests, and particularly at that time of year, is that our hope would be that particularly in infected, there would be tree sprays, that they would be, it would be less impact on our targets with anything that you use than what you would use once the trees really start leafing out and there's a lot more different insect activity.
Great. All right. Let's see. Peter asks, when I open the nest in winter, the contents appear to be powder-like. Does this nest have viable eggs in it?
Well, the nests actually contain the small caterpillars, not eggs. So if you open it up, you should be able to see small, and they're only about, less than a quarter of an inch, caterpillars, and they're quite hairy. And sometimes, like one that I opened up yesterday, had a lot of frass and leaf material in it, and it was at first difficult to see the caterpillars, but they were in there and they were alive. Yeah, so I think that the powdery stuff could be the frass. One of the things, I was surprised at how much frass was in the webs that I opened up yesterday. Because a lot of times when I've opened them up, there's not that much in the winter, because they're once they've closed them up. But I wonder, one thought that came to me is maybe depending on how warm the fall is, it can determine how much they continue to eat once they've formed the web. And that you could get a more accumulation of frass if they do a lot of feeding in the fall, versus if temperatures get cold faster and they stop feeding. But that's just a guess.
Great.
Yeah.
Well, I think no more questions are coming in so far. Lots of thank yous.
Okay. All right. Good.
That was great, Ellie.
All right. Well, thanks everybody for listening, and I'm actually hoping that we can, I've retired as well as Charlene, so I'm hoping that we can spur some interest in this topic. One of the things that has struck me is how little has been done on any insect silk except for silk moths. So I think that if people start to look into it, they will, and it's so critical to their survival and behaviors that it's something we should know more about.
Thank you, Ellie. Do you have time for one more question?
Sure.
This is,
Happy.
Helen asks if she can ask an off-topic question.
Okay.
If she's still here. Yeah, you're still here. Go ahead, Helen. She's asking who's taken on Myrmica rubra.
Oh, well, there are a couple of people who are working with Myrmica rubra. There's I know on a graduate committee for a person down at Clark University in Worcester, Mass, who is working with Myrmica rubra. So
Any news on control?
of Myrmica rubra?
Yes.
I haven't really heard any updates on what can be done more than the baits that are currently available, which I know are not ideal.
Thank you. Excellent talk.
Thank you.
One more question, Ellie? Apologies, I missed one.
That's okay.
Bob asks, I live in an area where our default oaks are bur oaks. A quarter-mile down the road, neighbors' trees are heavily infested and have been nearly completely defoliated the past 2 years. My trees, though, have mild infestations. Are there natural resistance variations among trees?
Well, there's actually Paul Schaefer, he found a lot of interesting things about host plants. Definitely, they impact everything from the size of the winter webs, the size of the caterpillars weight of the caterpillars going into overwintering, and their survival through the winter, as well as other factors. So I would think that that the host plant variation would have a large impact on browntail moth success. And Paul was able to show that in multiple aspects of the life cycle. So
Great. Well, thank you.
Can I add to that?
yes, please do.
So one of the other things that Maine Forest Service personnel observed over time is that the browntail moth will heavily attack certain trees one year and then shift to other trees in subsequent years because they're looking for healthy leaves for their young caterpillars to feed on and ones that haven't refoliated. So Bob may be lucky. He may have resistant trees, or he may just be lucky in that they haven't hit his yet.
Yeah, right. That's a good point, because because they are hitting the trees twice in a year, and if the overwintered caterpillars have completely defoliated the trees, then the tree may re-leaf, but those trees might be small, or they might have been hit so many times they don't re-leaf and refoliate, and so they find alternate sources. Yeah.
And the other thing is that if a tree refoliates, then those trees tend to have more phenols and chemicals in them to inhibit any more attacks from any kind of insect.
Right. Yep. Good point.
Cool. Great. Well, thank you everybody for coming. Again, appreciate your time, Ellie. Great to see you.
Oh, good to see you, Hillary. Thanks very much for inviting me. Charlene.
Hope to see lots of you on Saturday at the winter workshop.
All right.
Bye.
Bye-bye. Thank you.